Hello everyone. I'm from the University of Manchester, from the Department of Chemical Engineering, just over there somewhere. And I'm going to talk to you today about something a bit different from anything we've heard so far. I'm going to talk to you about using molecular modelling to provide understanding for water diffusion through organic protection coatings. So corrosion occurs across a wide range of both time and length scales. So we've had a lot of really nice talks already today. Look at things like this rusty bridge, or Stuart had a very nice, better picture of corrosion, but I couldn't find that one, so I'm stuck with a bridge. We've had talks on looking at sort of millimetre scale, so a blister or something. We had some really nice talks about sort of the micron scale, and this is actually one of Reza's images from a paper showing the different particles in a coating. But then corrosion itself happens at sort of the atomistic level, so the nanometer and sub-nanometer level. And it's actually this level that I'm going to be talking to you about today. So I'm concentrating on how water and ion diffusion, hopefully we can prevent that, or at least understand it through an organic coating. So I'm a computational chemist. So what I do is I construct realistic models on a computer of polymer solid systems, so it looks something like this. And from these, I'm able to identify the transport mechanisms, bottlenecks, energy barriers, and basically what's going on in these systems at an atomistic level. And the hope is that then I can provide fundamental understanding in what's going on in these real systems and things that understand experimental results that perhaps you need some sort of molecular insight into understand. So an organic protective coating, sometimes called a paint, is a formulated product. So it's really complicated in the short. So paints include things like binders, fillers, additives, all sorts of things. And their primary function is to protect the substrate underneath. So Simon gave a very nice talk about this yesterday. And this is some work done in Suscord, which we've heard a lot about in conjunction with AkzoNobel. But these paints are all formulated to the specific project, the specific application that they want. So the paint on aeroplanes is going to be very different from paint on cars, very different on the paint on the walls in here, very different for food packaging coatings. Basically, it's complicated at a molecular level, so what am I going to do? So the type of paint and organic coatings that I'm looking at tend to be found on these big sort of steel and iron structures like bridges. Obviously, I'm not going to simulate an entire bridge at an atomistic level because I don't think it could be done and it wouldn't provide anything helpful. But if we zoom in a bit, we can see this is what the actual protective layer looks like. So we have the structural metal underneath. There tends to be an oxide layer formed by some sort of surface pretreatment. Then you apply a primer layer of coating, maybe some interlayers, and finally a top coat. So even this is too complicated to look at an atomistic level. So what we do is we zoom in and we try and find out what are we interested in at the atomistic level. So to prevent corrosion, one thing these coatings can do is to stop ion and water transport through the system. So what I'm doing in my simulations is looking at how do these water and ions move through the coating itself. So to do that, I create this realistic cross-link structure and look at it with different water and ion concentrations in. And from this, I'm able to get out things like the diffusion of water and actually identify the water mechanism through the coating. So because this is very different from a lot of the other talks today, I thought I'd include a quick slide about what molecular modelling is. So basically, I represent, or the computer represents, each atom is a single bead with specific parameters. So my oxygen atom has a specific radius, interaction, and charge, which is different from my hydrogen atoms in this water molecule we're going to form. Then join these together using springs with a certain stiffness and length. In more complicated molecules, I also have angles and dihedrals, and there we have a molecule. So I then perform molecular dynamics, which is simply integrating Newton's equation of motion through time. So theoretically, you could sit down and do this on a piece of paper, but that's what computers are for. You wouldn't want to do that. So the system I'm looking at is an epoxy resin system formed of digiba and MXDA. So it's in a stoichiometric ratio. So this digiba can form 2 epoxyamine bonds. So here the epoxy is already open. Whilst each nitrogen in MXDA can form 2 bonds. What I do is I then cross-link them and I get a system that looks like this rather than the single water molecule that I showed you earlier. The nice thing about computer simulations is that we have highly controlled, we can control the independent variables separately. So I look at what happens with the temperature. You can also look at pressure. What happens with the cross-link density. So these epoxy resins form a three-dimensional network, which is how they work to protect the underlying substrate. And we can vary how many cross-links there are. And we can also look at the composition. So this could be the ratio. It could be what epoxy amine we have in there. But today, what I mean by composition is the amount of water we have present. So I've looked at systems between 0 and 5% weight water. We can then go out and we can measure some things. So we can get things like the pore size distribution. So when I talk about pores, they're much smaller than pores we've talked about before. They're on sort of the nanometer scale, sub-nano scale. Dynamics, so we can actually look at how the water and the polymer matrix move and get out some diffusion coefficients. And finally, we can look at the locations of water. So do we see sort of microphase separation? Do we see clusters of water? Where are the water located in relation to different atoms in the epoxy resin itself? And do we have any sort of hydrogen bonds forming? So forming the single molecules like water is trivial, but when we want to consider these three-dimensional networks, it's actually really difficult to do at an atomistic level. So I'm just going to talk through the procedure of how I make my cross-linked resin. So I have my two molecules. I have my digiba, shown here in this yellow with the red dots, which are the active sites, and MXDA, which is purple with green dots. And basically, I throw my molecules in a box and run it for a bit to equilibrate and get a well-mixed system. What I then do is I look in a sphere of a cut-off radius around each each digiba active site, and I try and find if there's any MXDA active sites within it. So there's one here. If there is, I then form a new bond. So I say a reaction has taken place, and this has now formed a cross-link bond. This bond is very high energy, so I have to run the system for a bit to relax it in a multi-step process. I then run another short molecular dynamic simulation to move everything around. So it looks like this. We've got our new bond still, and I essentially do the same. So I look around all the digiba reactive sites, see if there's any MXDA reactive sites that haven't reacted yet, here and here, form new bonds, and so on, until I reach the desired amount of cross-linking in my system. So if we do that, if we look down here, this is what the reaction is actually taking place. So you've got your digiba and your MXDA, and you form a bond. Once you've done all this to your desired amount, all the chemists within you will be like, hang on, your carbon's got 5 bonds there, that's not right. So we go through and we delete our excess hydrogens and update the charges on the carbons and the nitrogens. We then run a final equilibrium run, and then we have our cross-linked system ready to get all the information we want out of. So that's the reaction. So with simulation, there's a saying, you put rubbish in, you get rubbish out. So you need to validate your model to prove that what you're doing is actually what's happening in real life. So to do that, I validated my model using the density. So we get the same sort of density as you get measured experimentally and is in the literature. And also the glass transition temperature. So got the percentage crosslink here versus glass transition temperature for my work, which is these black circles, and 2 bits of experimental data from the literature. And you can see that they agree quite nicely across the crosslink range. So there's a slight deviation, and that's due to simply the finite system size. So I'm looking at much smaller systems, and I'm cooling at a much faster rate than the experimental system. So this is the type of agreement we would expect. So I'm very happy with that. So now I know my model is realistic. I can actually look at what we wanted to look at. So the first thing is the coating microstructure. So this is what my simulation looks like. You can just about make out the atoms they're in transparent here. And these blue regions are voids. So inside these blue regions, there's nothing. It's space, and it's where the water will go and where the water does go. So for all the systems I looked at, so all the temperatures, all the cross-linking, all the water percentages, they all had this similar structure of isolated pores. So there was no sort of continuous interconnected network. The nice thing about simulations is I can then quantify the pore size distribution at these different conditions. So the final thing that I'm going to say on this slide is increasing the water concentration, increase the proportion of large pores, which is to be expected because there's more water there, so your coating swells slightly. But all the trends that I'm going to tell you about in a minute were the same for all water composition, all the water content. So one of the main outputs is pore size distribution. So these show the pore diameter. So these are angstroms, so these are very small. So 10 angstroms is 1 nanometer, versus the probability of having a pore that size. For a low crosslink, 50% crosslink, or a high crosslink, so 98% crosslinked, which is the highest one we managed to achieve. And what you can see is that at a certain temperature, so if we look at this purple line, for the higher crosslinks, you get a higher proportion of larger pore sizes. So basically, as you form these crosslinks and these covalent bonds, you're causing the coating to expand, and you're actually creating more free space in the coating. With all the crosslinks, as you increase the temperature, so we go from purple to green to blue to orange, the amount of large pores increases, so your coating would swell slightly, but the behaviour is different depending on whether you've got a small amount of crosslinks or a large amount of crosslinks. So for small amount of crosslinks, you see this very big change in growth in large pores, whereas for the high crosslinks, you don't see much change, and that's because you've got these actual physical covalent bonds holding your structure in place. So as I mentioned, these pores are very small. We're actually only interested in ones that can hold some water. So the size of water is about 3.16 angstroms. So basically, anything this side of the graph, water won't fit in, so we're not really interested in it. But we want to know what's happening on this side of the graph. So because these pore size distributions, if we integrate the area under this, we can actually find out the probability of having a pore that's large enough to fit some water in. And that's exactly what I've done here. So now on the bottom we've got temperature, then I'm doing it for different cross-linking amounts. So at low temperature, what you see is that as you increase the amount of cross-linking, you actually have a higher probability of finding a pore that's large enough to fit water in. So this is what I was telling you about having the cross-links hold the system apart, so you've got more free volume and more larger pores. As you increase the temperature, for the 3 cross-link samples at the very high temperature, they're all essentially the same, and you see a very different behaviour for the uncross-linked sample. Basically, as you continue to heat it up, it just keeps expanding, 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 and you fit more and more pores in. So how does this relate to water diffusion, which is what we're actually interested in? Well, so in all cases, as you increase the temperature, the water diffusion rate increases for all cross-linking percentages. Makes sense. But as you cross-link your system, your diffusion coefficient decreases. So this is the opposite to trend to the one I just talked about with your free volume. So yeah, for 98% cross-linked, your diffusion coefficient is much less than your 0% cross-linked. So why? So the answer is actually it's governed by the polymer tray mobility. So it's the flexibility of your coating and the movement of the polymer that's sort of the key factor in the water diffusion, not the pore size distribution. So if we look at that, so now I'm instead plotting the percentage cross-link for different temperatures against diffusion of the polymer, not the water. And I will point out I've now gone to a log scale because otherwise you wouldn't see anything here. And you can see that for all of them, as you increase the cross-link, you get this dramatic decrease in the polymer mobility in the system. So for 450, you can see you get basically a 2 orders of magnitude difference in how flexible your polymer coating is as you cross-link it. And because we can see what's going on at an atomistic level, we can actually see the water diffusion mechanism through the system. So next slide should be a video. There we go. So I've selected at random a water molecule in green, and if we watch it, it will spend lots of time in one of these pores I talked about, and it will quickly shuffle through the polymer matrix facilitated by the polymer, spend some more time in another pore, and then shuffle around to another one. And all the water molecules do this separately. I just picked one at random. So in summary, using molecular modelling, we've managed to identify the diffusion mechanism of water through an epoxy resin coating. So it diffuses through isolated pores that's mediated by the polymer motion. The water diffusion increases with increasing temperature, but it decreases with percentage crosslink. And for the water percentages we looked at, up to 5% weight, it's pretty much the same. So this has impact in how you're coating curing, how effective it is. So you might have a slightly off stoichiometric amount, or you might not have cured it at quite the right temperature or something. So you might not have reached the really high percentage of cross-linking that you want. And also the environmental conditions, so like the temperature and the moisture content. Finally, I'd like to thank Suscord for giving me the opportunity to do this, and all my colleagues at Manchester, Sheffield, Liverpool and Axel Nobel. And thanks very much for listening to my slightly different talk today.
Thank you. Do you have any questions for Charlie? Yeah, do you want to go front here first?
That was a fascinating talk, thank you. I didn't quite understand how you decide what water is present initially, or do you put it in initially, or do you diffuse it in after the coating's been formed?
Yeah, so that's a very good question. So I put it in initially because it's much easier than trying to get it in once the coating is formed. But I have validated this by making the coating that way and then taking all the water out and checking that the coating that remains is the same as it would have been without any water there. So we're essentially assuming that we've got an equilibrium system here.
When you say an equilibrium system, that implies that you're in equilibrium with the water activity outside the coating in some way.
Yeah.
How does that control? I mean, presumably that implies you must know about the solubility of water in the coating.
Yes. So
how is that determined?
So it's not. I just picked 4 different water concentrations from the literature. So I know it can hold up to about 5% weight water.
So you sort of implied a solubility in the design of your coating, in effect.
Yeah.
Is that right? I have another question, if I may, and that is, a real paint is basically the painter coming along with 2 cans and mixing them together. I can't believe that you get a completely random mixture of the 2 components by doing that.
So I guess my answer to that is it's a question of length scales as well. So my scales are very small, so I agree you probably don't get a complete mix, but that's on a much bigger scale than the one I'm talking about. So I'd be talking about sort of the region where you've got this. If you have, say, a microemulsion, I'm talking about the region at the edge of that where these interactions will be taking place.
But to what extent does that, the poorly mixed parts of the coating, to what extent you think they actually control the water diffusion through the coating?
That's a very good question. I haven't looked at that because I've only looked at this stoichiometric one. Yeah. So to look at that, I'd need to go and do one with less MXDA in. And then
might be interesting.
It would be a very interesting way to expand this. Yeah.
Thank you. Thank you.
I think there was another question. There's one in the middle. I think there's one at the back as well.
Yes, thanks. That was very interesting. You mentioned at the start something to do with ions.
Yeah.
So you didn't carry on with that.
Yeah, no, they're the work in progress. They're the next step, the ions.
Yes, because you might like to sort of think about this idea of predicting D and I areas.
Yeah.
That you're aware of those and the way the conductivity varies.
Yeah.
Yeah, that would be the next step.
Yeah.
Certainly interesting. Thanks.
We have a question at the back. Yeah.
Charlie, do you deal explicitly with the hydrogen bonding between the pendant OHs and the water?
I've looked at them, yes. So from these systems, you have to use a geometric constraint to find them out. And yes, I have looked at them. I didn't have time to show them here, but I can. Okay. I can't remember.
You can't remember, okay.
But I do have that data somewhere. Yeah.
Okay. Thank you. I'm kind of asking this question because I think it's an interesting question, although I probably know the answer anyway. This is a blob of polymer in the middle of a gigantic piece of polymer. What happens if that blob of polymer moves close to a physical interface?
So this is actually what I really want to do next, is what happens when you put a surface there. I've got so many things I want to do next, is what happens when you put a surface there, because obviously the surface will affect how the polymer orders, and you might get some interaction with the surface, you might get some bonds forming. So the short answer is I don't know, but I would really like to find out.
Another more important thing is models are great, and I'm really impressed with this, but they need to be validated against an experiment. Can you tell us an experiment that might validate your model?
Yes, so there is, the word escapes me. PALS spectroscopy can help find the average pore size, which is the same as the free volume size I was talking about here. So we can compare them, and I think collaborators at Sheffield, so Stephanie, has been looking at neutron scattering. I don't think she's here today.
There's Patrick, who's
yeah.
Mr. Powell's over there.
Yeah.
So the other thing is kind of jump diffusion model you've done. NMR and sorry. So in your jump diffusion model, NMR data and quasi-elastic neutron scattering data have also looked at that kind of thing. I don't know if they've done it in your system, the GEBA, but they've done it in lots of other systems. So you can look at that jump frequency and the residence times too. So we'll chat about that later.
Yeah, that'd be great.
Okay, so we have a question from the online audience. Excuse me, when I read it. That's from Bob Luigi to everyone. Thanks for the nice talk, Charlie. How are you planning to look at ions? Are they going to be individual ions, or are they going to be solvated?
That's why I started with water. They'll have to be solvated, I think, is the short answer. But yeah,
so
ions
if you take again, it's a very important question. If you take an ion in water, it has a solvation sheath, several solvation sheaths indeed. What happens to that solvation sheath when you shove it into a polymer where the pore size only accommodates one water molecule?
Yeah. Well, these were the energy barriers that I was talking about right at the start. So hopefully, when I move on to ions, we can find out.
Okay.
I feel like I've answered all your questions with that's the next step.
That's the next step. A bit of that. Well, you do that in your next job, I guess. So congratulations to Charlie. He's got a job at Exeter. One thing that I think is important is, as an aside, as it were, your model, which demonstrated an increase in pore size by increased cross-linking, is something that we have found by over-curing particular types of coatings. So they're supposed to be cured after whatever length of time curing. If we cure them for longer and longer and longer, the coating expands, which was completely unexpected by AkzoNobel. But we can actually physically measure the expansion of the thing, and the only answer is it has increased pore volume, but it has increased corrosion protections. There's more water in there, but it's less transporting less.